everyone for coming today. So I'd like to introduce one of my undergraduate researchers, Ryan Miller. He's been working with me over the past year, collecting samples and analyzing data for an independent research project that he has been doing with me. So without further ado, Ryan Miller.
Thanks, Dr. Kleinschmidt. This is an image of the Los Angeles River directly west of here back in the summer of 2015. And this might be something that some of you are familiar with. And what happened here is there was a large ecological disaster where there was stored mining waste that through a series of events got released into the river. And this had tremendous consequences on the ecosystem and life in general in this ecosystem. And so what had happened is when we mine, there's naturally waste that gets produced that's associated with the mining process. And so people try to store that, but sometimes that doesn't go so well. So interestingly though, this type of mining waste can actually occur naturally, not just through artificial man-made mining. And so today I'm going to talk about a bioinformatic analysis of microbial communities associated with that natural waste. Before I start, though, I want to talk a little bit about microorganisms and their habitat diversity. So they can live almost anywhere. So these are images of places microbes are found, and they're all very extreme habitats. And so in addition to these type of habitats, they're also found with that mining waste. And those are the ones that we looked at in this research. So this is an image of how the waste becomes generated. So here's an example of a mountain. Now, when we go into mine, we cut into the mountain and we expose some of this rock. And the rock is high in minerals and heavy metals. That's why we're mining it. One thing that's found in this rock is something called pyrite. And so pyrite, it would be along this area. When it is exposed to oxygen, it becomes oxidized. Oxidized pyrite makes the water acidic, and this acidic water dissolves some of the heavy metals in this mine shaft. And it leaches out and contributes to this problem of mining waste. In the natural process, though, some of that rock that's heavy in minerals and metals can just be found laying on top of the ground. So you can imagine when it rains, we have the same process happening. So microbial communities can actually live in and use some of that waste as food products. And those communities are very specific. The local geochemistry and some other factors drive evolution of those communities to make them very, very specific. And so really that necessitates doing a study of these communities at each location. You can't just do a study here and apply those results over there. So for our study, we focused on the Alamosa River Basin. This is a map of Colorado for reference. When we zoom in directly west of Alamosa is our sampling location. And this is an image of where we sampled because we wanted to sample along a gradient to be able to observe differences in microbial community. So this red line, this is actually a small trickle that dumps into this yellow line, and that's a Lum Creek. And then a Lum Creek eventually dumps into the orange line, which is the Alamosa River. And so this really created a gradient of at the top, there was very low pH, harsh conditions, and then near the Alamosa River, the pH was more neutral and life would be more sustainable. These are actually images from those locations. So you'll notice above Lum Creek, there's a lot of this kind of burnt orange precipitate in the water. That's actually iron oxides that have leached into the water through this natural process as rain runs over some of this mining rock. And then you'll notice as we continue down this gradient and into the Alamosa River, the water becomes less concentrated in these iron oxides. Like I mentioned, microbes are able to live in those areas and metabolize things. And so what this kind of looks like is a microbe can take a sulfur group right here, and then it can combine that with a proton and reduce the sulfur. And when that happens, there's less protons in the water's less acidic, and some of those heavy metals that used to dissolve don't dissolve anymore and they fall out of the water. So that's an example of how a microbe can be able to help kind of clean up or bioremediate some of this waste. The specific aims of this project, our first aim was really to do a taxonomic profiling, to figure out who is there at a taxonomic level. and then to do a functional profiling and try to figure out not only who is there, but what they're doing metabolically. What metabolic contributions do they make to their specific community? This is our general project workflow, and we began with taking field samples. And so when we took these field samples, we collected soil and sediment samples from each area, and then at each of the sampling areas, we also took metadata that would include things like pH, oxidation potentials, water temperature, general stream flow, and things of that nature. And we started above Alum Creek in that red area, sampled along Alum Creek, and then along the Alamosa River. These are GPS coordinates of our actual sampling locations. And so you can see that we kind of established a gradient here starting above and continuing down the Alamosa River. Like I mentioned, we were interested in the geochemical gradient. And so up here I just included some general metrics that there was a low stream flow up in that area with high pH, or low pH, almost neutral pH with a higher stream flow in the Alamosa River. After we had done our field samples, we had to collect DNA. And so to collect the DNA, what we did is we took those samples and we took all the cells that were in them, the bacterial cells, broke them open, and then collected the DNA through a binding process. Once we had collected our DNA, we had to amplify a certain region of the genome for later analyses. And to do that, we use a process called PCR. And a PCR process basically allows you to make many, many copies of a certain region in a genome. And the region that we copied is actually the 16S locus. And this is a really, really useful area to copy when we do these type of analyses, because first of all, it's universally distributed in prokaryotes or bacteria. It's specific to prokaryotes. And then there's actually conserved and variable regions. And so we can design primers that bind to the conserved regions and then make copies of variable regions in between the conserved regions. The variable regions we can then sequence to distinguish what we might have. After our amplification process, we sent our samples to an off-campus location to be sequenced. Once we got the sequencing stuff back, the data, we had to process it. And to process it, what we were trying to do is we were trying to assign a species to our sequences. Now, with bacteria, this is a little bit tricky because they evolve so quickly that something that's not very similar can actually be the same species in the bacterial world. And so what we did is we took all of our sequences and we compared their similarity. And anything that was 97% similar, we considered as species. To those species, we then assigned taxonomic assignments. So if this was one group that was 97% similar, we took those and compared them to a database. Whatever they were similar to in the database, we took that taxonomy and assigned it to our groupings. After we did the raw processing and sequencing, we did a bioinformatics analysis. And bioinformatics is just a fancy way to talk about a computational analysis. Because when we got our results back, we had millions of sequence reads. And so you can imagine that a person really wouldn't be able to sort through those by hand and make meaningful conclusions. And so we were able to use computational techniques to help us along that process. Some of the things that a bioinformatic analysis can help you analyze include diversity analyses, composition analyses, and functional analyses. And these, we did do all three of these, but we started with the diversity analysis. And so there's two types of diversity analyses that we did and I will talk about today. The first is an alpha diversity analysis. So alpha diversity answers the question, how many species are there in a sample? So if you look up here, if we were talking about alpha diversity between these two groupings, the one on the right would have more alpha diversity than the one on the left, as it would have more different species. So when we took our samples, we basically broke them into two groups for this analysis. Instead of doing it with each individual sample, we broke them into a high stream flow and a low stream flow. And when we did the alpha diversity analysis, you'll notice that on the y-axis we have basically a metric of alpha diversity, and then we have our sequence samples. And so you'll notice that the high streamflow area had more alpha diversity than the low streamflow area. So this would suggest that there was more different species in the high streamflow, so in the Alamosa River, than the creek above Alum Creek and Alum Creek. After the alpha diversity analysis, we did a beta diversity analysis. In a beta diversity analysis, it answers the question is there a change in diversity between two samples? So for the first one, you'll notice we were just looking at each sample itself, but here we're comparing two samples to see if there's a change in diversity. So if we take these, you'll notice that they're the exact same. So when you compare them, even though they have high alpha diversity among themselves, they have low beta diversity compared to each other. So when we did this with our data, we grouped them similarly into a high and low stream flow. And you'll notice that this is a PICO-A analysis. And so the idea behind a PICO-A analysis, it's quite complex, but the basic idea is you take all your data and you reduce it to a single point. And each one of these single points represents a sampling location, and then their relative position on this coordinates indicates how similar they are. So things that are close together are similar, things that are further apart are not as similar. And so you'll notice that the high streamflow and the low streamflow samples were very similar to each other, but different when we compared them as a group. And so this would suggest that there's a difference in beta diversity between the high and low streamflow samples, and the beta diversity is driven by a change in species diversity. After our basic diversity analyses, we did a composition analysis. And this is an image of what that might look like. So there's a lot going on here, but let me see if I can explain it. So on this side of this circle, we have different bacterial phyla, or just taxonomic groupings. On the right side, this is actually our sampling locations. And this is along the gradient. So this is beginning at the very top and then continuing down to the Alamosa River, where the pH was low to near neutral. So you'll notice that these bacteria phyla are asymmetrically distributed. So the actual width of this line is relative abundance. So in certain, in this phyla, for example, there was a lot above, a ton in Alum Creek, and then hardly any in these other sampling locations. And so this was basically trying to point out that there was a difference in distribution of these phyla that could be driving the diversity. To kind of understand that better, we did a functional analysis, and this really aims to answer our second research aim: what are they doing metabolically? So what we can do is we can take our gene sequences and we can compare those to other gene sequences that we know the function of. If they're similar, then we can then make inferences about the function of our gene sequences. And so this is what we did for our data, and here's what we found. So again, we have a high and a low stream flow grouping, and these things up here, sporulation and electron transfer, these are different types of genes. And you'll notice that they are distributed differently. So in the high stream flow area, there's less genes that have to do with sporulation and electron transfer than there are in the low stream flow area. And so at the genetic level, this answers some of the questions potentially about what's driving that difference in diversity. We saw the phyla that were asymmetrically distributed, but there's also genes that are asymmetrically distributed. And it turns out that these two genes are actually involved in processes that bacterial cells carry out when they live in a harsh environment. So you can imagine in the low stream flow where there was a lot of the acid rock drainage, and a low pH, bacterial cells could need genes like this. So in conclusion, we were able to draw correlations between some of our metadata metrics and species diversity. So in areas where the stream flow was low and the pH was low, we observed low beta and low alpha diversity. In areas where there was a higher stream flow and the pH was more hospitable, we observed a higher beta and alpha diversity. Secondly, we were able to make some of those metabolic inferences like I showed on the previous slide about possible sporulation, electron transfer genes, and what contributions bacteria that live in this harsh environment make in order to help clean that up. Really though, like I mentioned with bioinformatics, it's really hard to analyze the data because there's so much going on. And so this data set, there's further potential to explore this data set and draw other conclusions. Future directions of this research, what we would like to do is, like I mentioned, we did the 16S marker analysis, and that's only a small section of the genome. What we would like to do is we would actually like to do a whole genome sequencing. Because for the metabolic inferences, it's kind of tough to make an inference from that very small section. But if we could sequence the whole genome, we should be able to make better inferences and get a better idea of what these things are doing metabolically. Lastly, and probably most importantly, what we're trying to do and the whole goal of this is to make correlations between acid rock drainage and acid mine drainage. So the acid rock drainage in these natural communities, the microbes are easy to understand, their interactions are less complex. So if we can understand how that works to bioremediate, we could potentially design a bacterial community that we could use in the mining process for these acid mine drainage communities that could then hopefully help bioremediate some of this mining waste. I thank Dr. Kleinschmidt for all of his help. This research would in no way, shape, or form be possible without his help. I also thank the Porter Scholarship Program for funding, and then these individuals for their contributions as well. Here are my references, and are there any questions?